specific nutrients interact with your genes. This field of study is known as nutrigenomics. In addition, we're going to look at how this affects metabolism and how you age in general. We'll also be discussing a few specific gene polymorphisms that can be readily genotyped, including MTHFR, FADS2, FUT2, ApoE, FOXO3, and more. The first step is a relatively inexpensive $99 23andMe test. You may be thinking to yourself, wait a minute, I thought 23andMe stopped generating health reports. You'd be correct, they did. However, they still give you the raw data, and this can still be very useful. The second step is a tool that's $5 called Promethease. Promethease will take your 23andMe raw data and match it to a very large database consisting of over 57,000 published single nucleotide polymorphisms. It'll then tell you what diseases you may be predisposed to, as well as how different micronutrients may have different absorptions and bioavailabilities, etc. My goal here is to discuss some of the more meaningful of the 57,000 gene polymorphisms, particularly those that may be influenced by modest dietary and lifestyle changes. For my own life, I've already found this information to be particularly useful in helping me and my family lower some of our disease risk to diseases that we may have been otherwise predisposed to. But before we get started, I want to cover a few of the basics, like what are gene polymorphisms? Gene polymorphisms are variations in the sequence of DNA of a gene that alters the function in a gene either in a good or a bad way. This is very different from mutations, which occur randomly because gene polymorphisms occur in a sizable percentage of the population, and they're thought to have been selected for a reason. Gene polymorphisms can lead to diseases like Alzheimer's disease, and also can lead to different phenotypes like eye color. For every gene that you have two copies of it, one from mom and one from dad. These are called alleles. In fact, if you were to look at your 23andMe raw data, you would see two letters which correspond to DNA nucleotides for that gene, one from mom and one from dad. In some cases, maybe only one of your parents has a specific gene polymorphism, and they pass that on to you. In that case, you would be heterozygous for that gene polymorphism, which may or may not have any measurable effect. In other cases, maybe both of your parents have a specific gene polymorphism which they pass on to you. In that case, you would be homozygous for that gene polymorphism, and that does have a measurable effect. Now that we have all the basics out of the way, let's dive into a few of the genes of interest today. The first gene polymorphism that I want to discuss isn't actually a gene polymorphism, it's a cluster of gene polymorphisms in the folate metabolism pathway. Folate, or vitamin B9, serves two very important functions. First, it serves as a precursor for the DNA nucleotide thymine, which means it's essential to make all new cells in the body, whether we're talking about new gut cells or new brain cells. The second important function is folate serves as a precursor to methyl groups, which are important for turning genes on or turning genes off. Most of the time, they're important for turning genes off. And this is known as epigenetics. This specific cluster of polymorphisms are associated with the gene 5,10-methylene tetrahydrofolate reductase, also known as MTHFR, and is associated with the reduced capacity to convert 5,10-methylene tetrahydrofolate into 5-methylfolate. 5-methylfolate plays a very important role for making those methyl groups that turn genes off and turn genes on. In addition, it also reconverts homocysteine into the amino acid methionine. This means that while people with these gene polymorphisms are able to make the DNA nucleotide thymine, they aren't able to efficiently convert dietary folate or supplemental folic acid into 5-methylfolate. For this reason, it's often associated with higher levels of homocysteine, which have also been associated with a whole host of vascular diseases, including coronary artery disease, stroke, and vascular dementia. Due to one or more combination of these polymorphisms in the MTHFR gene, approximately 40% of the population has 40% reduced functional efficiency of MTHFR enzyme. Approximately 20% of the population has a 70% reduction in functional efficiency of the enzyme, and approximately 10% of the population has between an 80 to 90% reduction in functional efficiency of the MTHFR enzyme. Despite the fact that a large percentage of the population has one or more combinations of this gene polymorphism, studies have shown that supplementing with 5-methyl tetrahydrofolate and methylcobalamin, vitamin B12, can successfully circumvent some of the shortcomings of these gene polymorphisms and lower homocysteine levels. Another common gene polymorphism affecting approximately 46% of the population is in the NBPF3 gene, and this affects vitamin B6 levels. People with this gene polymorphism have slightly lower vitamin B6 levels and may require that people increase their dietary intake of B6 and or also supplement with vitamin B6. Since we're talking about B vitamins, another gene polymorphism in the FUT2 gene affects the way vitamin B12 is absorbed in the intestines. People with this specific gene polymorphism need to take a sublingual vitamin B12 to ensure that they're getting enough vitamin B12. Let's move on to vitamin A, also known as retinol. We can convert carotenoids such as beta-carotene into provitamin A, also known as retinal. However, there are a cluster of gene polymorphisms associated with the BCMO1 gene that affect the ability of our gut cells to convert beta-carotene into retinal and reduce its ability between 30 to 70%. In fact, around 42% of the population has one gene polymorphism associated with its reduced efficiency, and approximately 24% of the population has another polymorphism also associated with reduced efficiency. People that have these cluster of gene polymorphisms in the BCMO1 gene can increase their dietary intake of animal products, which are high in vitamin A, or can supplement with the active form of vitamin A, not beta-carotene. But be careful not to take too much vitamin A, as it can be toxic at high doses. How about those omega-3 fatty acids? There are common gene polymorphisms in the delta desaturase gene, FADS, that affect the ability to convert the plant omega-3 fatty acid, alpha-linolenic acid, ALA, into eicosapentaenoic acid, EPA, which is often found in marine sources. This polymorphism can either increase or decrease the efficiency to convert ALA into EPA. Because this conversion efficiency is already so low, having the polymorphism that decreases efficiency even further is not a good thing and may affect how much fish people consume as well as what you supplement with. This is particularly relevant for vegetarians that often take ALA as their primary source of omega-3 fatty acids and may want to consider supplementing with another option such as microalgae oil, which has more EPA and DHA and doesn't rely on having to convert it from ALA. Let's move on to one of my personal favorites, vitamin D. There are a couple of common polymorphisms in the CYP2R1 gene, which encodes for the vitamin D 25-hydroxylase, the enzyme that is responsible for converting vitamin D3 into 25-hydroxyvitamin D, which is the major circulating form of vitamin D, which goes on to be converted into the active steroid hormone. People with these specific polymorphisms have a lower circulating 25-hydroxyvitamin D level and also have reduced enzymatic activity of the 25-hydroxylase. This has been associated with a higher all-cause mortality. Other studies have shown that people that have serum levels of 25-hydroxyvitamin D between 40 and 60 nanograms per milliliter have the lowest all-cause mortality and also have the longest telomere length. It's generally known that supplementing with around 1,000 IUs of vitamin D3 a day can raise serum 25-hydroxyvitamin D levels by around 5 nanograms per milliliter. This may not be the case for people with these gene polymorphisms, and other lifestyle factors may also play a role. What about choline? Choline also is a very important precursor, much like folate, to make methyl groups, which turn genes off or on. Well, we can make choline from phosphatidylcholine. However, 44% of women have a gene polymorphism in the PEMT gene, which does not allow them to make choline from phosphatidylcholine. This specific polymorphism does not respond to estrogen, which is known to activate the PEMT gene. For this reason, women with this polymorphism have to increase their dietary intake of choline. Egg yolk is a great source of choline. Let's move on to ApoE. There are a series of polymorphisms that make 4 different forms of ApoE, which encodes a lipoprotein that's made in the liver, which binds to cholesterol, transports the cholesterol throughout the bloodstream, brings it to tissues, and recycles it back to the liver. It's also made in the astrocytes of the brain, where it transports fatty acids and cholesterol to neurons. Approximately 25% of the population have a specific form of ApoE called ApoE4, which is associated with higher circulating levels of LDL and also is associated with a 2 to 3-fold increased risk for Alzheimer's disease. I'm currently researching ApoE biology both in the brain and in the liver, and I'm writing a paper on it right now for publication. I will be covering this topic in depth and comprehensively in a future video. And last but not least, let's talk about the longevity gene, FOXO3. FOXO3 is associated with longevity for several reasons. Flies and worms that have an equivalent of FOXO3 or the homolog have a 100% increase in lifespan. So worms go from living 15 days to 30 days. Mice that have the equivalent of FOXO3 experience a 30% increase in lifespan, and humans that have polymorphisms associated with making more FOXO3 have an increased chance of living to be 100 or being a centenarian. The reason FOXO3 is associated with longevity is because it turns on a whole host of genes that make you more resilient to stress. It turns on antioxidant genes. It turns on genes that repair DNA damage. It turns on genes that kill tumor cells, and it turns on genes that make sure proteins don't aggregate and clump inside of your cells. To sum it up, having more FOXO3 is pretty awesome, if you're lucky. In summary, knowing which gene polymorphisms you have is very useful, and that's why tools like 23andMe and Promethease are a great combination. Having this knowledge allows you to tailor your diet and lifestyle to your own genes and to optimize your micronutrient and macronutrient intake such that you may extend your lifespan when applicable. I'm Dr. Rhonda Patrick, and I'll catch you next time. Dr. Rhonda Patrick here. Today we're going to talk about sleep and insulin sensitivity. When most people think about insulin sensitivity and what regulates it, they think about macronutrient composition or calories or exercise or fasting. People don't usually think about sleep, when in fact sleep is a major determinant of insulin sensitivity. In fact, several studies have shown that sleep restriction over the course of several days can lead to acute insulin resistance in healthy individuals. Yet another study showed that sleep restriction for 4 hours a night, 2 nights in a row, reduced the insulin response by 30% and glucose tolerance by 40% in healthy individuals. And yet another study showed that just a single night of sleep restriction causes acute insulin resistance in healthy individuals. This really highlights the important role that sleep plays in glucose regulation. There have been several mechanisms that have been worked out, ranging from how the liver and the muscle absorb glucose to how the beta cells in the pancreas respond to glucose and in turn produce insulin. In a recent discussion I had with Dr. Matthew Walker, he discusses how sleep restriction causes the beta cells in the pancreas to stop becoming sensitive to glucose, and this in turn affects how much insulin is produced.
I think the blood glucose story and sleep is very, very well worked out now. It started with epidemiological studies where we started to see that people who were sleeping less than 7 hours were at significantly higher likelihood of either being diabetic or going on to develop diabetes. Many of them were already in what we call a diabetic state, or they had what we now call sort of metabolic disorder. And then the question became, well, is that associational or is it causal? So the next studies that happened, and this was work done back in, started in the 1990s by Ev Van Carter at the University of Chicago. Wonderful studies, took a group of healthy people, started to limit them to different doses of sleep for a week. 5 hours of sleep, 6 hours of sleep, 4 hours of sleep. And what she showed was that essentially after 1 week of short sleep, your blood sugar levels are disrupted so significantly that your doctor would classify you at that point as being pre-diabetic after 1 week of short sleep. And the way that they do this is what's called a glucose tolerance test, where you are fasted and then you are given this sickly sweet drink of glucose, and then they are measuring from your blood in the next 3 or 4 hours how quickly is your body able to dispose of that blood glucose. So what happens when you drink or when you eat a meal is that your blood sugar spikes, and you don't want that spike to stick around very long. If your body is healthy, it deals with that raised level of blood glucose very quickly, and it brings it back down very quickly. That's a healthy profile. That's what we call good glucose management. So how good is your body at disposing, essentially, of that glucose? And the way it disposes it is that cells in the body, including muscle cells, will suck up that glucose. And it's called your disposal index, or your disposition index, it turns out. So what she found, Van Carter, with her studies, was that, firstly, the way that your body knows how to absorb glucose and suck in that glucose is that there is another chemical called insulin, which is released by the pancreas and beta cells of the pancreas. And that insulin will instruct the cells of the body to open up special glucose channels to absorb the glucose, and your blood sugar drops, which is good and healthy. Firstly, what she found was that when you are not getting sufficient sleep, the beta cells in your pancreas stop being sensitive to the signal of high glucose. So the beta cells, which normally are listening for this spike in glucose, and as soon as they sort of hear, no, they're not hearing it, they're sensing it, but as soon as they sense the spike in glucose, they release insulin. And that insulin will drop your blood glucose. But those cells had become insensitive to glucose, what we call sort of glucose insensitivity. And so the beta cells of the pancreas stopped releasing as much insulin. It didn't release enough insulin to drop blood glucose, so blood glucose remained high. If that wasn't bad enough, we've since gone on to demonstrate, and you can do this with really clever studies, taking tissue biopsies. The cells of the body, including muscle cells and fat cells, their receptors stop being as sensitive to insulin. So firstly, you're releasing less insulin when you're sleep deprived. Right. But what little insulin you do release is not instructing those cells to open up the channels to take away the monsoon of the glucose that's flowing in the channels of the body. So on both sides of the glucose regulation, on the release of insulin to instruct cells to absorb glucose, and on those cells themselves to be sort of instructed by insulin, those cells became less sensitive to the insulin signal. And so as a consequence, your overall ability to deal with glucose became far more degraded, and blood glucose remained higher, which sets you on a profile of looking pre-diabetic.
Optimizing for good sleep is obviously very important, but there are other factors that also play a role. For example, exercise, particularly high-intensity interval training. Studies have shown that high-intensity interval training can actually ameliorate some of the negative effects that poor sleep has on regulating blood glucose levels. So the moral of the story is, get good sleep when you can. But if you can't, get a little exercise in there as well. I'm Dr. Rhonda Patrick, and I'll catch you next time.
Hi, I'm David Sinclair, and I'm a professor at Harvard Medical School, and our goal is to extend healthy human lifespan. You might remember a few years ago, my lab discovered a molecule in red wine called resveratrol that extended the healthy lifespan of mice. Well, what we want to do now, with your help, is to try a new molecule that we think could be many, many times better than that. In fact, it doesn't seem to just delay aging, but actually reverse it. What we'd like to do is test this new molecule called NMN in a mouse and see if it slows down its aging process and makes it live longer. And this would be the first step towards extending human lifespan. And you would be some of the first people to help and learn about whether this is going to be possible.
So I take NMN every morning.
What is NMN?
Good question. So
Shown that's like exercise in a bottle.
these are the things that we've been studying in my lab for the last 20 years. What are the processes that diet and exercise do for us that keep us healthy, and why does calorie restriction and intermittent fasting make animals live so much longer? And we think we've figured out a large part of how that works, and now we're mimicking that with molecules.
Is the idea that you can mimic it with molecules and it'll be as effective as intermittent fasting?
I think the molecules will be better. And not only that, when we add them on to a healthy diet and exercise in the animals, they do even better. It's like a supercharged mouse.
Now, when you add them on to the mice, do you also add them on with intermittent fasting? And is there an additional benefit?
We do. One of the first molecules infamous molecules that we are known for is resveratrol from red wine. That molecule, discovered it in my 30s, or at least linked it to aging. What we showed was that if you give it to a fat mouse, they're as healthy as a thin mouse. They live just as long. They didn't get heart disease and all of the other bad stuff. And it was additive. Same with exercise. If we give our latest molecule called NMN to a mouse and we exercise it, it'll run even further than it could with either of those alone. So it's not an excuse to sit around and just eat chips and watch TV. It augments a healthy lifestyle, gets you further than what you could get naturally. 5 in yeast. And what they do is they protect all organisms on the planet, plants, bacteria, humans, from deterioration and disease. They're like the Pentagon. They sense when we're hungry, sense when we're exercising, and they send out the troops to defend us. So when you put more of these genes into a yeast cell or a mouse, they'll live longer, between 5 to 20% longer. And so we think that these genes are responsible for the effects of dieting and exercise, which is great. What that means is we can now mimic that with molecules. So NMN is one of those molecules. So is resveratrol. You can think of resveratrol as the accelerator pedal for the sirtuin genes, and the NMN is the fuel. And without fuel, resveratrol won't work. So NMN is the gas in the car.
I've heard of resveratrol, but is NMN a new molecule? Is this commercially available?
Some people have started selling it on the internet.
The fucking internet.
It's related to NR, which is sold by a bunch of companies.
NR.
Yeah, nicotinamide riboside is a supplement that raises the levels of a molecule called NAD.
I feel like I should make a shopping list.
So get a pen. So while you're writing that down, Joe, so the sirtuins, okay, get this. So sirtuins need NAD to work. Without them, they don't work. In fact, if you don't have NAD in your body, you'd be dead in about 30 seconds. It's a really important molecule. But as we get older, we lose NAD. So by the time you're 50, like I almost am, you have about half the levels of what you had when you were 20. So that's not good. And these sirtuins, they don't protect the body without high levels of NAD. So what NMN does, and this other molecule called NR, which both you can get on the internet, they boost the body's levels of NAD back up to youthful levels again. And if we give them to mice, these molecules to mice or even to worms or yeast, they live longer and they're super healthy.
Now, what level, like how many milligrams are you taking of these things?
NMN is something I get from myself. I'm not selling anything. So I take a gram of NMN in the morning. Based on clinical trials, it's been shown that will raise NAD.
With or without food?
I take a little bit of yogurt that I make myself at home just to
Look at
settle
you.
me. I've been doing this for a while, and I only start doing stuff when I see it work in animals first. So take the yogurt, mix in some resveratrol. Resveratrol is great, but it's really insoluble. It's like brick dust. So in the yogurt, it'll dissolve. Take another half a gram of resveratrol.
And how much? Half a gram?
Yeah. It's a powder. I have a few kilos left over from clinical trials in my basement. So yeah, that's going to last me a few decades. And then I also take at night some metformin, which is probably the most radical thing that I take, which is a prescribable drug for diabetes.
Metformin?
Met.
M?
M-E-T.
And prescribable drug. But you don't have diabetes.
I do not.
But you take it for
preventing cancer, heart disease, Alzheimer's, and aging.
Can you spell it? M-E-T?
F-O-R-M-I-N. And so out of studies of 10,000 people and more, it's been shown that people who take metformin, even if they have diabetes, are protected against other diseases of aging, even frailty. And so most scientists, if you ask them in my field, will say, yeah, metformin is likely to extend your lifespan. It's just that the FDA doesn't let you have it for aging, because aging isn't a disease yet. I also take supplements, and in fact most of my colleagues are in the field of aging, or anti-aging as people call it. So I take NMN every morning.
What is NMN?
Good question. So let me take a quick step back.
Sure.
So about 20 years ago, Lenny Guarente and a team of us at MIT discovered a set of genes that controls aging in yeast cells, just brewer's yeast, what you find in beer and bread. And those genes are called sirtuins, and there are 7 of them in our bodies, 5 in yeast. And what they do is they protect all organisms on the planet, plants, bacteria, humans, from deterioration and disease. They're like the Pentagon. They sense when we're hungry, sense when we're exercising, and they send out the troops to defend us. So when you put more of these genes into a yeast cell or a mouse, they'll live longer. That could be trillions of dollars. Just $50 trillion just in this country alone that could be spent on figuring out how to solve global warming, better education, the environment, saving the 1/3 of species that are becoming threatened. That's why I think tackling aging isn't a selfish act. It's probably the most generous act that I could give the planet.
That's an interesting way of looking at it. People buying supplements, and the supplements have other things in them. And what are you drinking? Do you drink mostly water? Do you consume caffeine?
So with NMN, which gives me a boost, I think, caffeine's a little too intense.
Really?
I find that. So I'll have a cup of coffee in the morning, but that's it.
One a day?
Yeah.
Wow.
Yeah, just to kickstart
How do you live?
It's hard.
Kind of
It's
living
really
is
hard. Sorry. It's all going on, and a regular brain that isn't trained would have a hard time.
Yeah, I would imagine. I'm so shocked that you only drink 1 cup of coffee a day with that kind of schedule.
So the NMN is good.
Do you take NAD in an IV form?
No.
No. Now NAD in supplemental form though, in terms of orally ingested NAD.
So if you give NAD to an animal or a cell, it's taken up really poorly. NAD is a big molecule. NMN is a smaller version of NAD that gets into cells quickly. So that's probably a difference.
So it's the same benefit, but it's easier consumed orally?
Well, I think so. I mean, we're still at the cutting edge of figuring out what's true and what isn't, but NAD is thought to not be well absorbed in the body as compared to these other smaller molecules that the body then turns into NAD once it gets in.
They look great.
They do. So my father is a prime example of that. So he's in Australia and he's been taking care of his body since he was in his early 40s. Probably a little too late, but still. He's been taking some NMN and some metformin for a while, resveratrol. But he's been active. That's the key, I think. And then, so that combination, so he's now just turning 80 this year. You would think that he's 30. If you didn't know his age, he's running around. My mother passed away from cancer, so he's dating women. He's out every night. He's traveling the world. Now, this is the future for people in their 80s. He started a new career.
At 80?
It was at 76.
What's he doing?
He got bored. So he retired at 67, thought he had another 10 years of good life, and he kept going and going and going. His friends are getting frail. He's still active. So he went back and he's working at Sydney University on the ethics panel for clinical trials and other studies. And that's what you want older people with wisdom and knowledge to do, to give back.
That sounds good, too. Like, for you get your dad hopped up on all these awesome new drugs. Then you get him working on the ethics panel for clinical studies. And then you get him to give you some death row patients so you can try it on, right? Ethics.
You're onto me.
Yes, I know what you're doing now, man. You're wiggling the system a little bit with your
keep it in the family.
Hyperactive, super healthy dad.
Right. Well, I don't recommend anything even to my family, but they end up demanding it. My brother was pretty upset that we weren't giving him any.
Well, it seems like it's working out really well for your father. I mean, so
Seems
metformin,
like it,
the NMM, and what else is he taking?
He's also on a statin and he exercises. That's the main.
So he's on a statin as well because he has the same predisposed condition, you know.
Right.
Yeah. That's an awful condition, man. That I mean, one of the things that I was talking to my doctor about, he was saying that there are people that just have high blood pressure or higher blood pressure or higher instances of heart disease in their family, and it's just a really unfortunate genetic issue.
It is. But fortunately, we're able to tackle heart disease pretty well these days with blood pressure drugs and cholesterol drugs. There are some side effects, no question. But what we're talking about with these longevity drugs that are in development is that, sure, you can be prescribed this medicine for your Alzheimer's or for your liver disease, but as a side effect, it'll keep the rest of your body healthy as well, protect you against cancer and all these other things. So that's what's so radical about what we're doing.
Situation.
Well, it is. And so we have 3 dogs. My wife runs a therapy dog organization.
Barbara Streisand had her beloved dog Samantha cloned. That crazy bitch. Look at her.
2 copies of one dog.
Jesus Christ, that's so strange. How about just get a new dog, you fucking nut?
So we might be nuts in my household because we're giving our dogs NMN.
Oh, and how's it doing? How old's the dog
so
now?
So good. The oldest dog is 9 and he's still doing fine.
Does he look different?
My wife says so. These are anecdotes. I'm not going to publish them, but so ours is a therapy dog, and he has to go to hospitals and nursing homes. And if he has NMN, according to my wife, he can't be a therapy dog because he's too excited. He's running around, jumping around.
Really?
So that's anecdotal, but that seems to fit with what others have experienced too.
Wow. If like 15, 20 years from now, your dog's still chasing balls and people are going to come over your house. Hey, Dave, what the fuck's going on with your dog, man?
Right.
Is that the same dog?
That's Charlie, yeah.
How come Charlie doesn't have gray hair anymore, man?
Right. Well, people ask me.
You don't have any gray hair.
No.
Yeah, man, that's weird, isn't it? You don't have any?
That's weird, right?
How long have you been on this stuff?